Great. Thank you for that introduction, Alex. So the title of my talk is Clinical-genomic Correlatives in Urothelial Cancer, and focus here on ctDNA, as Alex mentioned. So I'll talk about the genomics of urothelial cancer, some background on cell-free DNA. I'm going to touch a bit on sort of the infrastructure that enables our research, and specifically related to genitourinary biobanking here in Vancouver. I'll talk a bit about an ongoing project I'm leading looking at ERBB2, maybe more commonly known as HER2, as a predictive biomarker, and then some future directions for our urothelial cancer program. So urothelial cancer arises from the urothelium lining the urinary tract, and I think news to all of you that these tumors can originate anywhere in the urinary tract, so the renal pelvis, the ureter, the urethra, and the bladder. But most of them do arise in the bladder, and most of these tumors are also of urothelial carcinoma histology, although there is variant histologies that can occur both in isolation as well as concurrently with the urothelial carcinoma. This is the fifth most common cancer diagnosis in Canada, 10th globally, and by far the largest risk factor for urothelial cancer is tobacco smoking. But there are a number of hereditary genetic factors that can influence your risk for urothelial cancer development, Lynch syndrome being one of those. There are two sort of main stages here that I'm going to talk about. So non-muscle invasive bladder cancer being everything TIS, T1, TA, T1, and then marking in sort of an aggressive transition would be the muscle invasive bladder cancers. This is T2 to T4. These, while they represent only about a quarter of initial diagnoses, frequently metastasize and are obviously the main cause of urothelial cancer related death. Now, in terms of treatment options, when we look at the advanced urothelial cancer setting, platinum chemotherapy was the mainstay treatment for decades. But in the last few years, we've had a number of emerging therapies for sort of defined patient subsets, and this includes several PD-1 and PD-L1 targeted therapies. The first FGFR inhibitor, which is erdafitinib, as well as antibody drug conjugates. And as of last week, it looks like now we're going to have antibody drug conjugate IO combination. With this explosion of treatment options, the key question now is therapy sequencing and which patients are going to be most receive the most benefit from which therapy, right? So there's a number of options, but when do you give them to whom? And that's where molecular stratification is really going to be critical to optimize both selection and sequencing of all of these therapies. And this is particularly important with urothelial cancer because it is highly molecularly heterogeneous. So there's a lot of really great studies looking at primary tissue from both non-muscle invasive bladder cancer and muscle invasive tumors. These are well characterized, and we know that there is heterogeneity at the genomic level in terms of the driving events in each patient being different, at the transcriptomic level with different RNA subtypes that have prognostic implications, and at the cellular level with some of the different histologic variants that I mentioned previously. The mutation rate in urothelial cancer is the third highest of all solid tumors. So it's
just one moment. Okay, good.
So it's just below lung and melanoma. And as I mentioned, there's sort of this heterogeneity. So what's driving one urothelial tumor in terms of the genomic backbone would be very different than, say, another one. The molecular landscape of patients with metastatic urothelial cancer, comparatively unexplored until very recently, and this is largely due to the challenges of obtaining sufficient number of metastatic biopsies. So we have a ton of treatment options. We have a very heterogeneous group of tumors. We need biomarkers to direct the therapy choice. How do we do that? Gold standard, obviously, is to perform biomarker testing on the archival formalin-fixed tissue from the diagnostic biopsies or the surgeries. Particularly in the metastatic setting, however, this may not reflect the current disease state, right? You're talking about potentially years, maybe a decade or more, time difference, different intervening lines of therapy, potentially allowing for some sort of evolution of the tumor. And metastatic tissue biopsy is not routinely performed, especially outside of academic medical centers. Molecular stratification is clearly going to rely on having relevant tumor material. So what do we do? And this is where liquid biopsy comes in as a complementary or alternative biomarker source. Liquid biopsy is a term that encompasses a lot of different assays. It includes cell-free DNA. So what is cell-free DNA? It's DNA that's released from cells that are rapidly proliferating and turning over. It's fragmented due to the nuclease activity in your blood. And in the blood plasma, the majority of this cell-free DNA is actually coming from your white blood cells as they turn over. That makes sense. But in the cancer patients, some of that is coming from the tumor itself. So this is how we distinguish circulating tumor DNA from cell-free DNA. Circulating tumor DNA is the subset coming from the tumor. And the amount coming from the tumor really varies. So it can be undetectable, even in a patient with oligometastatic disease, or it can be more than 90% of all the cell-free DNA that we're seeing. So across solid tumors, circulating tumor DNA is a really promising biomarker source. It enables real-time, sort of rapid profiling of the tumor genome. You can look at all sorts of different genomic events, including amplifications, mutations, translocations, as well as epigenomic markers. And there are applications for this throughout the disease course. So you can use circulating tumor DNA early in the disease for screening and diagnosis. You can use it for detection of minimal residual disease after curative intent therapy or surgery. And you can use it serially in later stage disease to monitor treatment response or clonal evolution. The type of assay that you're going to apply with ctDNA really varies based on the clinical or scientific question you're trying to address. So there are two main categories of assays, those that detect circulating tumor DNA and those that characterize it. Detection assays, these are often used early, like in non-muscle invasive or muscle invasive bladder cancer perioperatively for surveillance. They're generally tumor informed, meaning that you have tumor tissue that you've sequenced and you're looking for specific mutations that you already know are present from the tissue. And these are highly sensitive assays. What I'm interested in mostly is assays that characterize circulating tumor DNA. They perform a comprehensive genotype, and this is done without any information from the tumor tissue. You don't need tissue. This is best applied in the advanced disease setting where circulating tumor DNA makes up the majority of the cell-free DNA. And it really does independently inform on the tumor genome in real time, comprehensively. And we can use this, for example, to identify FGFR alterations to give a patient erdafitinib without any knowledge from their tissue sample. So we showed sort of the first time that broad ctDNA profiling is really feasible in advanced urothelial cancer. We performed targeted sequencing with a panel that we designed that encompassed 50 genes. We looked at 51 patients that had either locally advanced or metastatic bladder cancer. And we found that not only was the amount of cell-free DNA you were getting higher in the patients with metastatic cancer, the amount that was tumor-derived was higher, and that makes sense. So the majority of patients that had metastatic bladder cancer had detectable circulating tumor DNA, sufficient for us to do comprehensive genotyping assays. This was not the case when we looked at patients that had muscle-invasive disease. Generally, the amount of circulating tumor DNA was very low and insufficient in most cases for this sort of tumor-agnostic approach. So from there, we then wanted to build a larger cohort focused on the patients that had metastatic urothelial cancer, specifically where we could perform this comprehensive genotyping. And we assembled a cohort of 104 patients with plasma cell-free DNA, at least one sample, as well as in many matched tumor tissue. This is all sort of published work, so I'm just going to touch on a few key findings as background. What we showed here is that the amount of circulating tumor DNA that you were able to detect prior to first-line systemic therapy initiation was prognostic for overall survival, or predicted overall survival. And so in this setting, we had 71 patients where blood was collected prior to treatment, and we split them on the median into low and high ctDNA categories. That was at about 5%. We found that those that had low circulating tumor DNA, which indicates low tumor proliferation, had much better prognosis. And when I built a multivariable model incorporating routine clinical prognostic factors, this was independently predictive. The next most important finding from the study was that the circulating tumor DNA mutational profile really did match what we were seeing in the tumor tissue. So we were able to reproduce most of the information. And that's shown here on the left. On the right is a more recent study, which kind of confirms the level of concordance that we were observing. So the mutation profiles are very similar, as you can see from the top in the blood and the bottom in the tissue. The copy number profiles are very similar from the scatter plot. Now, importantly, not every mutation is concordant. We don't expect it to be, but this has implications for targeted therapy eligibility. And finally, we were able to detect clinically relevant biomarkers in the circulating tumor DNA. These included ERCC2 mutations, which are known to be associated with improved progression-free survival and platinum chemotherapy. This included FGFR mutations and fusions, which again are key eligibility criteria for erdafitinib, and ERBB2 alterations, which I'll talk about a little bit more in detail. So now that you've got the relevant background, I just want to pause and talk about the biobanking in Vancouver that really has facilitated this type of retrospective research. Getting the patient numbers to dive into some of these molecular subtypes, getting samples and clinical data, this is really facilitated by both local and international collaborations. And here locally in Vancouver, we have 2 different genitourinary biobanks that have both provided samples for the projects I was just talking you through. So the first you're probably all very familiar with. This is led by Dr. Black. This is the Prostate Center Genitourinary Biobank. You may not be familiar with the BC Cancer Biobank of predictive biomarkers in GU cancers. So this is founded by Kim Chi and Alex Wyatt in 2016, specifically for the collection of plasma for cell-free DNA analysis. But through this biobank, we also have consent for a lot of detailed clinical records and the archival formalin fixed tissue at any hospital within the province. And since 2016, we have collected more than 4,100 blood samples from over 2,900 patients with genitourinary cancers. And obviously quite a large subset of those come from patients with urothelial cancer. Now, biobanking, it's a huge endeavor. There's a lot of invisible work that goes into this. So we have a huge clinical research network. We have 4 co-directors, myself, Dr. Marie Strahr, Dr. Chi, and Dr. Wyatt. We have clinicians, nurses, site coordinators, and clinical data collectors, project manager. This requires coordination throughout the province at all the different BC Cancer sites. We contract Life Labs to collect the blood and FedEx to ship it to our central lab here at the Vancouver Prostate Center for processing. And then here in the Vancouver Prostate Center, we have a team of pathologists, technicians, bioinformaticians, and obviously a number of research scientists that draw on this rich resource. In addition, I've spent a large part of my postdoc working on some aspects of our data management plan, including working with the team to develop a dedicated REDCap database for collecting all of the clinical data. So we have a REDCap database for prostate cancer clinical data and a separate one for urothelial cancer clinical data. And these are very large, robust resources just on the clinical data side alone. I have also developed a laboratory information management system on the backbone of Open Specimen. This is allowing us to track the tens of thousands of derivative samples that come from all these specimens. So from that rich resource of biobank specimens, we now have spent many years working on a number of innovative sequencing methods that we apply to that plasma, as well as the FFPE tissue sample from the same patients. We have custom next generation targeted sequencing panels. For bladder cancer, we're on our 4th iteration, constantly updating with new targets as evidence emerges and new targeted treatments become available. We also do perform some whole genome sequencing of circulating tumor DNA, as well as transcriptome sequencing of the tissue. This slide is courtesy of our bioinformatician, Andy Murtha, and it just goes to show that for as much thought has been put into the logistics and infrastructure of the biobanking, the wet lab assay development, there's similarly a tailored bioinformatic pipeline where we take the sequencing data we generate, and we can generate from that germline and somatic mutation calls, copy number alterations, and rearrangement data. So now with the background, as well as some of the infrastructure considerations, let's return sort of to the overarching objectives of our urothelial cancer program. So The main goal here is to determine the prevalence of prognostic and predictive biomarkers through the use of circulating tumor DNA, to explore the associations with the genomic alterations we're seeing, these molecular features, as well as the clinical data that we have and the patient outcomes, and then to look at mechanisms of evolution and acquired therapeutic resistance across serial same patient samples. So we have all of the samples from, their initial TUR of the non-muscle invasive disease, all the way through to serial blood collections in the metastatic setting. Under that umbrella, these are two projects we currently have ongoing. One is looking at FGFR and the other at ERBB2. The FGFR study is a national project led by Dr. Bernie Eigel for patients with metastatic urothelial cancer that are eligible to receive erdafitinib. That's the FGFR targeted therapy. The patients enroll in this study. They have blood collected, which is sent to us for processing and genomic analysis. And then they have their tissue sent for centralized standard of care tissue testing. And we're going to be looking at the concordance of the tissue to liquid biopsy to see if they could be used together to better identify the group of patients that would benefit from erdafitinib. And this study should be closing by the end of the year. But what I'm going to talk about today is the ERBB2 project that I've been leading, which is a retrospective analysis of the biobank circulating tumor DNA, looking at mutations and amplifications in ERBB2 and comparing that to immunohistochemistry and fluorescence in situ hybridization. So ERBB2 is the gene, HER2 is the protein. It is a cell surface tyrosine kinase growth factor receptor that is overexpressed, amplified, or mutated in several cancers, including urothelial cancer. It is targetable with monoclonal antibodies, small molecule inhibitors, and I think most excitingly, antibody drug conjugates. Selecting patients to receive HER2 targeted therapy is generally done from immunohistochemistry, looking at protein expression levels, and there's scoring systems both for breast and gastric cancer that are approved. You can also do in situ hybridization to measure the gene copy number. This is often done as a confirmatory assay based on the IHC results. Of course, the other option is next generation sequencing of the tissue or the plasma ctDNA to identify the causal mutations or amplifications. In urothelial cancer, historically, no benefit was observed with trials looking at HER2 targeted therapies. And this includes single agent trials as well as chemotherapy combination trials. And they are successful in other cancers, breast cancer being the prime example. So why not urothelial cancer? That's sort of the question the field's been asking for quite a while. And I would posit that potentially the molecular heterogeneity that I described earlier is a key factor here. And that really highlights this need for biomarker-driven patient selection. The other aspect of this is that we have really promising clinical trial data emerging right now with the HER2-targeted antibody drug conjugates. And so the other factor in terms of sort of success in the urothelial cancer space is improved therapeutics. So you have better biomarker selection, patient selection, and then improved therapeutics. And together, I think these two are going to enable HER2-targeted therapy to really shine for patients with urothelial cancer. So there are a number of HER2-targeted antibody drug conjugates in clinical trial. I list a few here. You'll notice that sometimes the antibody can be used in different combinations with different payloads. So you have your antibody, your payload, and your linker. They have different drug-to-antibody ratios, as well as the linker itself can be cleavable or non-cleavable. And this matters in the fact that if you have a cleavable linker, what that indicates is that the payload can reach cells that maybe do not express the target antigen. And so in a HER2 low expressing tumor where there's some heterogeneity, perhaps then this antibody and payload could cleave and target cells that are not actually expressing HER2 and lead to response in HER2 low tumors. So there's a number of ongoing clinical trials looking at this both as a monotherapy as well as in combination with other agents. We showed previously that in ctDNA, you can detect ERBB2 alterations, and that when we looked at the matched tissue of these patients, they generally were concordant. The alterations in the tissue were linked to high mRNA expression levels, and the alterations in the circulating tumor DNA, in a handful of patients, we looked at the immunohistochemistry and FISH staining and found that those were also concordant. So this is really promising in fact that we might be able to use ctDNA as an adjunct to tissue in this setting. So that's the basis for the project that I'm currently working on, where we're looking at ctDNA to characterize ERBB2 as a predictive biomarker, specifically for patients that have metastatic urothelial cancer. The objective here really is to explore this nuance in ERBB2 status. I think we often think of biomarkers as positive versus negative, but that doesn't really represent the full picture, because you have to really think about the assay limitations and how did you arrive at the threshold for positivity versus negativity. So there's a lot of considerations going on before you even get to that sort of positive negative readout. And how does that impact clinical relevance and targetability of these different mutations and amplifications? So this is going to be done looking at serial, tissue, and circulating tumor DNA samples. And then we'll benchmark the results from our plasma circulating tumor DNA against the standard of care archival tissue testing, which is immunohistochemistry and in situ hybridization based. Really, the goal here is to optimize the utilization of HER2 targeted therapy for precision oncology approaches. So we had 413 plasma cell-free DNA samples from patients. This represents 236 unique patients. And we profiled these with our targeted sequencing assay. We also profiled patient-matched leukocyte samples in every case, so this provides a germline control. Of those 236 patients, 181 had at least one sample that was positive for circulating tumor DNA. So had enough circulating tumor DNA for us to do this genotyping assay. And then of those 181, we identified 36 patients that had ERBB2 alterations. So this is 26 patients with a protein altering mutation, 17 with an amplification, and 7 of those patients had both a mutation and amplification in the same sample. And that equates to about 20% of the patients having an ERBB2 alteration detected via ctDNA. One of the first things that we did was check whether the genomic landscape we were seeing in the patients that had an ERBB2 alteration was consistent with what we were seeing in the patients that did not. So we saw that regardless of their ERBB2 status, these metastatic urothelial cancer samples showed frequent aneuploidy, very similar tumor mutational burdens, and what I'm showing in the plot here, most importantly, is very similar circulating tumor DNA fractions. I say most importantly because circulating tumor DNA fraction, as I showed you, is prognostic in and of itself. So we want to ensure that there's no bias between these different groups based on ctDNA fraction. Moreover, ctDNA fraction limits the type of alterations we can detect. So we need more circulating tumor DNA to detect an amplification than we do to detect a mutation. So it was really reassuring that we saw that these subgroups were very similar. And that there was no sort of bias as a result of different ctDNA fraction between the patients with and without an ERBB2 alteration. So then diving into the patients where we did detect an ERBB2 alteration, this oncoprint shows at the top, for each column is a different patient. At the top plot, we have their tumor mutational burden. Below that, we have the proportion of the cell-free DNA that was tumor-derived. Then we have the number of copies of the ERBB2 gene, and you'll notice that some patients have more than 50 copies of the ERBB2 present, while others, sort of on the right-hand side, dip down to sort of what you would expect diploid to 2 copies. We have base ploidy, so whether or not we detected aneuploidy in these samples, structural variants, and you'll notice they cluster in the patients that have the highest copy number on the left-hand side, and so I'll talk about that in a moment. And then the specific mutations that were detected in these patient samples. From this, we noticed immediately that there are patients where there's an oncogenic mutation and an amplification co-occurring in the same tumor. However, no patient had more than one oncogenic mutation. So those were mutually exclusive. If you had one oncogenic mutation, that seemed to be enough. However, non-oncogenic protein-altering mutations, those did co-occur, and they were often linked to high tumor mutational burden. So what this suggested to us is that these are passenger alterations. It really does highlight the fact that they're probably not activating mutations in ERBB2. the oncogenic mutations that we found, these are known hotspots that are activating and drug sensitizing. They lead to constitutive activation of the receptor tyrosine kinase. We found that the mutations tended to be clonal. So here, clonality refers to the portion of cancer cells that contain the mutation. And you can imagine that if the majority of your tumor has this mutation, then that's probably a very good target. If it's only present in a handful of cells in your tumor, or one lesion out of a patient with many different metastatic lesions, it's probably not going to be a great target. But in the patients that had oncogenic ERBB2 mutations, 14 out of 17, that was clonal. Non-oncogenic mutations were more likely to be subclonal, again indicating they're probably passenger alterations. Now, mutant allele-specific imbalance is a phenomenon where the mutant allele is selected for and amplified, whether through copy gain or through loss of the wild-type allele. And this is linked to targeted therapy sensitivity pan-cancer. It really does indicate that alteration is driving the tumor. And so we wanted to look whether or not mutant allele-specific imbalance was present for the patients that had an ERBB2 mutation, and whether it was more common maybe here than in other HER2-positive cancer types. So what we found is the minority of patients, only 3, actually exhibited mutant allele-specific imbalance, and that's indicated in the box here. So purple are copies of the gene that are containing the mutant allele, and then gray is copies of the gene that are wild type. And you can see in those 3 patients, all or the majority of the copies of ERBB2 contain a mutation that is activating. But in the rest of the patients off to the right, you don't see that phenomena. So I do hypothesize that maybe this is going to be different cancer to cancer type and may underlie why HER2 targeting is more successful, say, in breast cancer, if there's often a sort of a mutant allele imbalance in that case. So I'm looking at getting some data sets together to compare. So switching from mutations to amplifications. I mentioned previously, we talk about biomarker status, positive, negative. We talk about ERBB2 amplified or not. But the reality is this is not binary. This is a continuum. And so we have patients with 2 additional copies of ERBB2. We have patients with more than 130 additional copies of ERBB2. So what is the biological relevance of a patient that has only 5 extra copies versus 50? And in the red box here, I've highlighted 3 patients, all of whom have about 20% circulating tumor DNA, so similar detection ability, similar prognostic implication, yet one has maybe 7 copies, the other has about 10, 11, and the other one has about 40, 50 copies. So there's quite a big difference in the number of copies of ERBB2 that each patient can harbor. We wanted to see now, what is the target of these amplifications? So ERBB2 is located on the Q arm of chromosome 17. And what you see in this plot is the entire Q arm, and then the number of copies of that region of the chromosome. And so you're sort of going along and it's relatively diploid, and all of a sudden there's a spike where the number of copies of that region in the chromosome gets very, very high, and then you go back to being diploid. Well, that spike is ERBB2. So if you look at the inset plot, you can see a zoom in. Each trace is a different patient. Where the amplification sort of boundaries are varies patient to patient, but all of these are converging on the ERBB2 locus and trying to encompass that gene. So what this indicates is this is not a chromosome arm level event. This is really targeting ERBB2. And there's a number of possible mechanisms for this high level focal amplification that we're looking into. When we look at the structural rearrangement data, we found that this really did support those amplification boundaries. So this is separate data that we get from the sequencing where we can look at sort of where are predicted structural variants. And these are going to occur as the number of copies increases. There's just going to be sort of some messiness in terms of the chromosome. It's not a perfect cut and paste. And so in green and orange at the top, you can see these traces indicating different structural variants. These are 2 different patients on the left and right. And in both cases, those structural variants line up exactly with where we see the sort of stepwise focal tandem duplication phenotype boundary. One of the other questions that sort of arose to us when we were looking through this data data was whether or not some of these cases may be extrachromosomal DNA. So extrachromosomal DNA are circular fragments, often containing an oncogene. They're sort of, as the name implies, they're not on the chromosome. They're circular fragments outside of the general chromosomes. And they enable this rapid increase in genomic heterogeneity, transcriptional upregulation, because you can have a lot of copies of these ecDNA fragments, and they may not segregate sort of in a Mendelian fashion on cell division. And so what we saw is that actually in about half of our cases with an ERBB2 amplification, there was at least one other oncogene that was amplified to a similarly high level. And this is the copy number plot of one patient that really, struck this idea home, where CCND1, another known oncogene in bladder cancer, ERBB2, the gene that we were studying, and BRCA2 were all amplified to the same level. And those are the three red dots you see there. BRCA2 is a tumor suppressor. This is normally lost, not gained. The majority of these gained copies are non-functional. They harbor a truncating mutation. So why? Why are there so many copies of BRCA2 in this patient's genome? And so we hypothesized that potentially it's actually in the same extrachromosomal DNA fragment as the oncogenes. It just got carried along as a passenger. And we're looking at doing whole genome sequencing and running a tool called Amplicon Architect, as well as doing co-fluorescence in situ hybridization in order to sort of really dive into whether or not these are co-localized ecDNA. Yeah, there's absolutely. So there's a really interesting paper that came out a couple months ago looking at actually this in breast cancer, and they sort of coined a new type of amplification mechanism where there's a bridge forming. And so they showed in breast cancer that this happens, where you have multiple oncogenes on different chromosomes sort of forming bridges during cell replication and then breaking into these ecDNA fragments. It was a really elegant paper that made us think that this was something that was feasible here. The other hypothesis that I've had floated to me is that they're actually not in the same ecDNA fragment. They're in separate ones somehow that are co-localizing during cell division. So the mechanism for that is kind of unclear as well. So one of the big strengths of our research program is that we can look at serial samples in patients with metastatic disease where metastatic biopsy is not feasible, and then explore temporal heterogeneity. So how does ERBB2 genotype change over time? Obviously, as I mentioned, the patient selection is currently based on immunohistochemistry and in situ hybridization. And these are well-standardized, relatively cheap assays. So if we want to propose a next generation sequencing-based assay to identify eligible patients, we really need to prove that we're going to find patients that would be missed by standard of care tissue testing. And what our sort of look at the literature has shown us is that while the mutation rate is relatively constant across disease stage, the amplification rate is not. So it increases as disease stage increases and aggressiveness. It's higher in UTUC, it's higher in metastatic disease. In our data set, we've looked at this evolution and found a few examples, and this is one of them that I want to highlight to you that really epitomize this idea that your ERBB2 genotype is not static. So this is a patient where we had blood drawn at metastatic diagnosis, again at the completion of their first-line chemotherapy regime, and at the completion of their maintenance immunotherapy. And what you see in the dots are individual mutations we detected at each of those time points, and the number of copies of that mutation. In pink is an oncogenic mutation in ERBB2 that is found in one copy, so balanced at baseline, again at the end of their first-line chemotherapy, but by the time they've completed their maintenance immunotherapy, the copy number has jumped. And this is actually one of those mutant allele-specific imbalance cases. So the mutant allele has been selected for, and that copy has been amplified. We actually have another ERBB2 mutation in this patient, not shown in blue, that at baseline is subclonal, and by the second and third samples is completely eliminated. So there's a clonal shift in this patient as they progress on treatment, eliminating the non-oncogenic mutation and amplifying the oncogenic one. So the conclusion, sort of interim for this project, is that a ctDNA-based approach is feasible to determine nuanced ERBB2 biomarker status in metastatic urothelial cancer, and that consideration of it as a non-binary biomarker is really going to be important to refine our understanding of response to HER2-targeted therapy. We are currently analyzing the patient-matched formalin-fixed paraffin-embedded tissue specimens. So we'll be doing DNA sequencing with the same panel. We'll be doing tissue transcriptome sequencing, and the clinical grade IHC and FISH is being done by BC Cancer, and I'm expecting those results probably this week, to be honest, for the first half of the patients. And then, as I mentioned, we're going to look a little bit more into the mechanisms of amplification, including whole genome sequencing and co-FISH for the ecDNA oncogenes, and correlate these molecular variables that we've identified with clinical outcomes for these patients. Now, these patients did not receive HER2-targeted therapy. This is a retrospective analysis. So one of the next steps will be to look at this prospectively in sort of a trial situation, comparing ctDNA to tissue in patients that are about to receive a HER2 ADC. So with that, I just want to talk for the last few minutes about where do I see our urothelial cancer program going from here. A lot of what I've talked about is retrospective research, and obviously the next stage then is prospective validation, whether that be for the HER2 alterations, that FGFR study that we have currently ongoing, or this sort of trial idea that's currently in planning phases with CCTG, which we call PROMOTE. And this is a multi-center phase 2 umbrella study, which is going to evaluate targeted therapy in patients with advanced urothelial cancer based on circulating tumor DNA biomarkers. We have a similar study currently ongoing with CCTG for prostate cancer, where we've screened over 500 patients. And so the idea is that they would have their blood collected, sent us centrally. We would run our targeted sequencing assay, identify eligible mutations and amplifications and structural variants, and then they would have a number of different treatment arms that they could receive based on that molecular profile. But obviously, single features in isolation, they're rarely strongly predictive or prognostic. I think there's a big shift in our field towards multi-omic biomarkers. One example being for the prediction of cisplatin response in muscle-invasive bladder cancer. And so these multivariable models incorporate clinical pathological features, genomics, transcriptomics, potentially even epigenomics and proteomics. One avenue for this, I think that's really, really prime, is immunogenomics to predict immunotherapy response. You need a composite biomarker that incorporates tumor cell information, immune cell information, and tumor microenvironment features. On the left is a really fascinating paper by Marcus Eckstein's group, where they looked at the immune phenotype of metastatic tissue from urothelial cancer patients to predict immunotherapy response. And what they developed was sort of a classifier based on the preservation of MHC staining by IHC. And this was able to predict immunotherapy response if you did the MHC staining from metastatic tissue. But when they used the same patient's primary tissue, it didn't work, right? So the primary tissue does not reflect the metastatic disease state. And really fascinating is that there are now tools available where you can do HLA loss of heterozygosity analysis from whole exome sequencing. And so you may not need to do immunohistochemistry. You may not need to even have the tissue if this is applicable from plasma ctDNA. So this is something that I've been working on with Nicolette Fonseca and Aslan Munzer, but basically running some of these tools on whole exome of ctDNA to predict HLA loss of heterozygosity, which you could then use in a multivariable model to predict IO response. So the work on the right is showing a patient where you can clearly see in the ctDNA loss of HLA-B in the tumor. Sort of another avenue for expansion of our program is epigenomics and transcriptomics, specifically into the field of fragmentomics. So ctDNA fragmentomics, they can give us information on the epigenome and the transcriptome because the spacing of the nucleosomes really does sort of read out in the NGS data that we have. And so this is, I think, we're all familiar with Cameron Herbert's Nature paper from last year, looking at this in prostate cancer. where they showed that you could look at sort of depletion around the androgen receptor from fragmentomic modalities. And so we're looking at whether we can do this in bladder cancer to identify sort of active transcriptional regions, and also whether we can do this at lower resolution, because this was done with deep whole genome sequencing, which is quite cost intensive. So one thing that our group is looking at is whether this can be done sort of on a more targeted level. But just to say that there are options beyond genomics, we can look at epigenomics and epigenomic marks, maybe without even doing another assay like a 5-HMC assay. But I think probably what you're most going to be interested in here, given the audience that I have, is urinary tumor DNA. So the urine supernatant and the cell pellet both contain tumor-derived DNA, and this is really optimal for the non-invasive detection of tumor DNA in the localized disease setting. There's obviously a lot of potential here, both at diagnosis, risk stratification and prognostication, monitoring response to systemic therapy, and surveillance post-treatment, including detection of minimal or molecular residual disease. One of the questions I get asked a lot is, should I be collecting urine? Should I be collecting plasma? I think this really needs to be driven by the tumor stage and the clinical question. So the assay that you choose is going to vary based on what you're trying to understand. I think urine tumor DNA here really is applicable anytime you have an intact bladder in patients with localized disease. Circulating tumor DNA from plasma is most applicable in patients that have a muscle invasive disease through to metastatic. And again, what I was speaking to earlier is if it's muscle invasive, you're going to need to do a tumor informed assay where you sequence the tissue and you do some sort of MRD assay. If you're in a metastatic setting, then you can do a tumor agnostic comprehensive genotyping approach. I think what's really beautiful is that in this sort of space of muscle invasive bladder cancer, where they're going to inform on two different things. You're going to have urine tumor DNA informing on what's going on with the localized disease. And if you collect plasma at the same time, you can look at circulating tumor DNA for the detection of potentially micrometastases, clonal evolution. That's the problem, right? So all of these, like any other urine-based test, there's a number of them out there. The sensitivity and specificity are great in high grade and not so great in low grade. So keeping that in mind is the limitation of the approach. What we're establishing here right now with Dr. Black and our Finnish colleagues, Matti and Ella and Jussi Nicola, they've really pioneered some methods in Finland for looking at urine tumor DNA. And the wet lab workflow, the bioinformatic workflow, these are really similar to the established ctDNA pipeline that we have here in Vancouver right now that I've just been showing you all of the results from. We can use affordable, targeted next generation sequencing panels, and most importantly, the Streck preservative. So all of the blood collections that we do, the reason that we're able to have them shipped at room temperature via FedEx and processed, days after blood draw is because of the Streck preservative. There's other brands out there that you can buy the specialized tubes that stabilize cell-free DNA. Well, Streck now has a urine preservative that we're using that you collect your urine. Well, actually, you'll see supplied me with some here. You collect your urine and then this little blue bottle goes in and preserves the urine at room temperature for up to 7 days. So that enables us to collect wherever you like. In Finland, they're doing at-home collections and then mailing them through the standard Finnish post. But you can imagine, you can collect in any OR and then just have them kind of collected by the biobanking staff at any point. So this is really fabulous, and I think it's going to be an interesting aspect of our research program where we can start to address questions in the muscle invasive and non-muscle invasive space that we couldn't address with plasma ctDNA. So just to summarize what the future directions are for our urothelial cancer program: expansion into urine tumor DNA, prospective evaluation of our ctDNA results in biomarker-directed clinical trials, integration of ctDNA into composite prognostic and predictive biomarkers, and expansion beyond just genomic characterization into epigenomics and fragmentomics. I'll just acknowledge that this is a huge effort. There's the Wyatt lab and a number of us who focus specifically on urothelial cancer within the Wyatt lab. It's not just prostate. Our key team members at BC Cancer, as well as just other VPC members and international collaborators. So thank you so much for your time and Any questions?